All right, thanks to ACD for the invite to present on behalf of the Analytical and Structural Chemistry team in early oncology at AstraZeneca. The title of my talk is Pharmaceutical Discovery and Development Analytical Workflows. So here at AstraZeneca, we are a global science-led biopharmaceutical business, and our innovative medicines are used by patients worldwide. Patients are at the center of everything we do, and it's most important to have the right target, the right tissue, the right safety, the right commercial plan, the right patient population, all while delivering these priorities in an organization with the right culture. The drug development process takes years with huge amount of information being generated. This involves thousands of individual experiments and tests, hundreds of reports, and terabytes of data. There'll be many analytical chemists working on a project supporting medicinal chemistry, all the way up through process chemistry and early development. Often we need to communicate this information with multiple stakeholders. These are the chemists, the formulators, toxicology and safety reps, and operations teams. We are always working with companies like ACD in order to develop how to figure out what kind of solutions we can implement to make our data more accessible. So since we generate and handle so much data, we need to make sure that it's findable, usable, and in a central and easily accessible location. In addition to this, we want to take all of this data and leverage it to help speed up our design, make, test, and analyze cycle. And a huge effort that we're working on here is this is our automated drug design platform. And so having all of our data accessible and easily read by this system is key to a lot of our future plans. So let me introduce you to the Global Analytical Database, which is powered by ACD. So first, we retrieve our data at a particular instrument. Once it's acquired, we have pre-processing data. This adds it to the spectral database with a link to the raw data and towards long-term archives. It extracts the metadata that describes the sample. So we have the sample ID, the chemist, the particular project, and where it was collected. It also extracts other metadata, which are NMR parameters or chromatography conditions, and other parameters as well, such as temperature or how much sample was used. It also associates a structure retrieved by the sample identifier, and this generates a SMILE code and includes the description of the structure. So even though archiving and capturing the data across sites is different by name or date or location, it doesn't matter once it's all in the GAD. So let's take a look at an example of accessing the Global Analytical Database. So here we can use ChemSketch to do a similarity search. For instance, we can search on a specific pharmacophore, and the database is not only for NMR data, but it's also for MS and chromatograms as well. This really helps a lot with patent writing. It saves everyone a lot of time. There's no need to contact an analyst to retrieve the data, and it's all on a single platform, searching to get all the data that you need. NMR Workbook is great for everyday processing and capturing spectra in ELN. We have created unique printouts based on the template used for the team. For instance, we have reports for chemists, and then we have reports for NMR specialists, and each one is tailored to give all the information that's needed. This was very easy to implement across all sites to produce a report that was standardized and captures all the data that the chemists were used to seeing in their reports. And after a couple training sessions, the chemists really adopted this format. And this was a huge benefit for change management. So for moving from one software company to another, it was relatively seamless. And now the spectra processing and reporting is actually much faster and easier for the chemist. As I mentioned earlier, we also use it with mass spec and analytical UPLC. And for every compound, we have a retention time and mass recorded. And again, the report templates here, they look very nice. They're easily incorporated into our notebooks and other archives. So As you can see, the data access is a huge aspect when you're generating large amounts of data. And in the past, if we're working on a paper or publication that was done a few years ago, it used to be a very tedious process to get the data. So often it would take a number of mouse clicks and writing an email to the analyst and waiting several days for a reply to get your spectra that you were looking for. So now once it's in the GAD, we can do this in just a few moments, and then with as little as 10 mouse clicks, get all of the data that you're looking for. And so this is really important to have all this data at our fingertips when we look to implement machine learning in AI models as well. In addition to automatically processing and storing data, we have worked with ACD to create scripts for reporting in specific workflows. So on the left here is shown an example of open access use for chiral analysis. And with the click of a button, we can calculate the enantiomeric excess for a particular reaction. Recently, we have expanded the script for QNMR. So now it automatically populates the milligrams of standard and sample. And this is information that's extracted from the metadata that's input into ICON NMR. So now when we receive our process spectrum, we only have to integrate the peaks, and within a couple of minutes, we can get our NMR strength report done. So this is simple, it's accurate, and it saves the chemist a lot of time. Moving on, I want to present a nice case study. For covalent inhibitors, we need to find a balance to find the right warhead. If it's too reactive, there'll be toxicity issues, and if it's not reactive enough, we'll have potency issues. So we usually need to monitor the reactivity of the warhead by LCMS-based kinetic studies. In a retrospective analysis, we demonstrated that the NMR chemical shifts can rank the reactivity of covalent warheads. All this was possible because we had this NMR data at our fingertips. And moving forward in the project, rather than doing the time-intensive LCMS studies, we were able to predict the reactivity of these warheads just by tracking this proton chemical shift. So this study was possible because we had rapid access to all the NMR data, and this shows the power of having all the NMR data in one central, easily accessible location. And what's most important here is that these studies are time dependent, and we're able to get this data quickly back to the team to inform the design of new chemical warheads. The next case study I want to present is looking at the NMR signature of flexibility. So when creating drug-like molecules, we often want to introduce rigidity into a molecule. So here, if we call up a set of NMR spectra from a particular series, we can compare the rigidity within the linker region. So when a CH2 group is diisostereotopic, this indicates that we have rigidified the linker region. So looking at our flexible linker, we see that we have a single peak for Z, X, and Y. So these CH2 groups are flexible and they're moving fast, so we just have an average single for each CH2. In our rigidified linker, we begin to see two sets of peaks for X and Y, indicating that that area of the linker is now rigidified. So this method is sensitive, but it's not always specific. There could be some false positives, so if we introduce chirality without necessarily rigidifying, but nonetheless, you can see it's a quick and easy way to assess the flexibility within a linker region, and the data is easily accessible, which is the most important part. So lastly, I want to highlight a decision-making pyramid. We have all of our data and information. We interrogate and build up our knowledge to make informed and our best decisions. Data processing and storage will be the foundation that we need to set in order to achieve our goals in predictive analytics and predictive insights. So for instance, if we train our models with the right data, we can get all of the predictive information before a molecule is even made. And this enables us to make the right molecules for the right target. And lastly, I just want to acknowledge all of the colleagues at AstraZeneca and our collaborators from AZ. And with that, I'll be happy to take any questions.